Hello everyone. We were left with about 37 slides before Wednesday, and about 1/3 of you were able to make it to class, and for the remainder I hope to make this as close as possible to the presentation that I delivered live today in class. We left off with the antimetabolites. We finished methotrexate and pemetrexed, and we have a good number of antimetabolite drugs left to discuss before we're finished with them. And we do have some other DNA modifying drugs to talk about, as you can see when looking at these slides here. So we're just going to wrap it up with the DNA modifying drugs for this presentation, and then we'll continue with Dr. Bomarity for the next lecture, which will be live. Okay, as I mentioned, let me try to move this out of the way here. As I mentioned, we left off with the antimetabolite drugs. Okay, we're going to move on to the purine-like drugs. Okay, hopefully you can see my cursor here. I'm using a software I've never used before. Up the top here is the structure of a real purine or endogenous purine. The drugs are in the second row here. We have 6-mercaptopurine and fludarabine. Okay, these drugs, there's a lot of complex things we could say about them. We could get heavily into the medicinal chemistry, but I don't really want to worry about that. The way they work basically is that they were designed to mimic the purines down here in DNA and RNA. So 6-mercaptopurine, the drug, resembles guanine and or hypoxanthine, which is only found in transfer RNA. And then we have fludarabine, which is an adenine analog. So I have the corresponding endogenous base from DNA or RNA down here. Okay, in these drugs, I would say the pharmacogenomics are at least as important as the mechanism by which they work. Okay, so next slide we have first 6-mercaptopurine. I'm going to get into a little more detail on this one. 6-mercaptopurine can be considered definitely a prodrug. It needs to be converted to its active metabolite, that is 6-thioinosine 5' monophosphate, or just say you can call it TIMP. It does that intracellularly, the conversion. And the metabolite, this TIMP metabolite, is going to inhibit what we call de novo, or from scratch purine synthesis. So some purines are synthesized absolutely from scratch, and some of them are actually scavenged from the extracellular space to be recycled. But we're talking about the from scratch synthesis that we're going to block. And really most of these drugs are going to interfere with DNA synthesis. Okay, as I mentioned right here, the pharmacogenomics to me are one of the most important aspects of this. Okay, and hopefully you'll remember this slide from gastrointestinal pharmacology when I taught with Dr. Balesta. Okay, we have, this is the metabolic route here. So what I have in gray here, which you're not going to see obviously in your handout because it's black and white, what I have in gray here is all the inactive portions of this pathway, the inactive components. 6-mercaptopurine, yes, it's absolutely inactive until it's converted. Has to be converted into TIMP. I don't want you to memorize this enzyme HGPRT. That's not critical. What is important to me is that you understand that TPMT is highly polymorphic in humans, and if someone happens to have a low function or non-functioning TPMT, you can imagine what would happen. This portion of the pathway will be blocked, reduced, and then the only portions or the only pathways left are on the far left here and the far right. So we're going to have shunting into these pathways. That means we're going to get some more active metabolite. Unfortunately, with chemotherapy, which is what this is, the therapeutic activity, an increase in therapeutic activity usually goes along with an increase in toxicity, because the pharmacotherapeutic effect is identical to the toxic effect or mechanism. So those have to go hand in hand for all this chemotherapy. And I mentioned in class today, for those of you who are listening to this for the first time hearing this lecture, I mentioned in class today that TPMT, even though you see it down here as well, in forming another active metabolite, if this is polymorphic and it's a reduced function polymorphism, you'll just have accumulation of this other active metabolite. So the ultimate effect is with TPMT dysfunction, you have TIMP toxicity. You have toxicity of 6-mercaptopurine. The other important enzyme is xanthine oxidase, perhaps not as much because of polymorphisms, but because of a drug interaction. And since you guys weren't there in class, I'm not going to try, I'm not going to try to do questions through this mechanism or this presentation and try to have you guys sit there on your own and figure it out. So I'll just tell you exactly what we talked about in class, and that's xanthine oxidase, which you already know, you should know, is that it's inhibited by allopurinol for gout. In that, if you block that, just like TPMT, if this gets inhibited, we're going to have shunting of 6-mercaptopurine to the active metabolite and more toxicity. And I also mentioned in class that febuxostat, which is the newest xanthine oxidase inhibitor, should do the exact same thing, should have the exact same toxicity issue. And I'm sure it must be listed that way in micromedics, but I haven't looked. You'll probably hear about allopurinol more because it's been used for a longer period of time. But since that blocks xanthine oxidase, the febuxostat or Uloric, it should do the exact same thing, cause toxicity. And some physicians, if you look down the bottom here, some physicians recommend not even using 6-mercaptopurine if someone has a TPMT dysfunction or reduced metabolic activity. Some say maybe start tenfold lower, so there's not a specific guideline here. The idea is that you need to monitor carefully, either not use it or use it at a very low dose, and if you're going to titrate up, do it extremely gradually. Okay, so this is the question. The answer would be allopurinol, obviously, is going to be the drug interaction there. Okay, next slide. Switching gears. I'm probably going to move a little bit quicker than I did in class. Okay. The next one would be pyrimidine analogs. If you don't remember from biology, pyrimidines are just the other bases in DNA and in RNA. So we have cytosine, thymine, and uracil as the endogenous bases in DNA and RNA. And then we have the cytosine analogs, cytarabine or ARC and gemcitabine. We have capecitabine. Okay, that doesn't appear to resemble a specific base, and then 5-fluorouracil, which is of course a uracil analog. Okay. Let me try to get this out of the way. Okay. 5-fluorouracil that's the focus of this slide right here. 5-fluorouracil. You've already seen this. If you remember from yesterday, you should remember, or from Tuesday, depending on when you watch this, maybe yesterday. Depending on that, the 5-fluorouracil basically can compete with DUMP. Okay, let me rephrase that. This 5-fluorouracil is the drug that we can administer IV, and that is going to mimic. Now, please bear with me. Go down here. This 5-fluorouracil is going to mimic endogenous uracil. If you see what that does, so that undergoes transformation into deoxyuridine monophosphate here. If we administer 5-fluorouracil, the only difference is, we don't have this hydrogen. Instead, we have the fluorine group. So it undergoes the same metabolism and it forms fluorodeoxyuridine monophosphate with this F dump right here. And what you have endogenously is that the normal chemical is going to compete with this fluorodeoxyuridine. And once fluorodeoxyuridine binds to thymidylate synthase, it doesn't get metabolized. It actually locks it in place or prevents it from functioning. So the whole point is that when we have 5-fluorouracil, we ultimately have thymidylate synthase inhibition, which reduces the amount of thymidine monophosphate, and then and then in turn diphosphate triphosphate, and this is necessary for DNA synthesis. So we have reduced DNA synthesis. Okay, so it's a lot of words, a lot of verbiage just to say that we're reducing DNA synthesis. Okay, so these all do it by their own sophisticated mechanisms, but it's the same idea. I mentioned in class this slide is not something I want you to commit to memory. What I meant was really this diagram down here. You don't need to know the details at all of this diagram. What I think it's important to understand is that capecitabine is a prodrug, and when it's metabolized, it is converted into 5-fluorouracil. So I want you to realize that the pharmacology is going to be the same as 5-fluorouracil, except the manufacturer of this, which I'm actually forgetting who it was, but the brand name is Xeloda. The manufacturer is claiming that this Capecitabine, when it gets into the cancer tissue has more thymidine phosphorylase than healthy tissue, which means we're going to form more active metabolite in the cancerous tissue and supposedly have preferential anti-cancer activity and less non-specific toxicity. And I haven't actually looked at the studies to see how true that is, but that's the claim. Okay, so maybe we could ask Dr. Manning or read some articles on that to determine if that's the case. Okay, moving on to next slide. The next slide here, as I mentioned, one of the most important things is going to be the pharmacogenomics. There's about 6 different proteins that can be basically have pharmacogenomic differences between patients. Remember, we had KRAS, and now we had TPMT, and now there's DPD. So 5-fluorouracil is going to be metabolized by DPD, or dihydropyrimidine dehydrogenase. Okay, so nearly 80% of it is hepatically metabolized by this enzyme. To keep a long story short, some patients have a dysfunction in this enzyme, or they have a mutation in the gene, rendering DPD less active. Okay, and these patients can then experience toxicity. Okay, I mentioned in class that I had quite a lengthy talk about the difference between phenotypic and genotypic testing for DPD, and I mentioned when we think of phenotype, we generally think of traits, and a lot of the traits we think of might be appearance. If you look back at Bio 121, maybe you saw that the peas were green or shriveled, or they were nice and plump and firm, or yellow. So all those traits, those are phenotypes. But what I'm referring to here's the enzymatic activity. The phenotype we're talking about is how active DPD is. So that's the function. Or we could look at the gene itself, DPD gene, and see if that's mutated. Phenotypic testing is actually, it can be done with 5-fluorouracil, and what they do is to be, ethical and gentle with the patient. Instead of giving them 5-fluorouracil, they can give them a test dose of the endogenous compound, but they're going to administer that, right? Exogenously, they're going to administer uracil, just plain uracil instead of 5-fluorouracil. They give plain uracil, and they attach a radioactive compound, or actually, I think they make one of the carbons, C14. In that way, it's not really toxic, the radioactivity. It's very mild. But what they could do is track the metabolism to see how fast does that uracil get metabolized. And that is a really strong predictor of how well 5-fluorouracil is metabolized. So if they find that there's slow uracil metabolism, then they could probably decide to not give 5-fluorouracil or give a lower dose. Okay, and I would always claim that phenotypic testing is ideal compared to genotypic, because phenotypic is answering the exact question that you want to know the answer to, which is what is the function, how well is it functioning. Genotype, well, that's going to tell you there's a mutation, but you don't know exactly how the function is going to be of an enzyme until you actually measure it. Okay, but genotypic testing is effective as well. Okay, and you could look for the specific mutations that reduce the function. And this is just the metabolic pathway. I don't want you to memorize that. It's just kind of for your information to see how that's metabolized. Okay, but this slide is meant to show you, basically some clear evidence to indicate how important this metabolic route is for 5-fluorouracil. If you look on the left here, we have a normal clearance of 5-fluorouracil in a patient with no mutations in DPD. On the right here, we have 2 curves, but this is the same group of patients. In the bottom curve here is just a correction for renal function. But what they're showing you here is that in a patient heterozygous for a non-functioning DPD protein, what I mean is if one of the alleles for DPD is mutated so that it doesn't produce functional DPD, we could actually get a 50% reduction, right, or even a more than a 50% reduction in the total body clearance of 5 fluoro uracil. That means just one mutation, just one allele, not both genes, but a heterozygous mutation can cause a very significant reduction in the clearance of 5 fluoro uracil. So what I said down here was, imagine if we had a homozygous dysfunctional DPD, we might have an extremely slow clearance and extreme risk for toxicity. And the reference down here, if you're interested in reading that. Okay, so that wrapped up the purine and pyrimidine analogs. Okay, and the only goal there is to get them incorporated into the DNA to hopefully block DNA replication. And they just do that all by kind of different mechanisms, but essentially they're replacing our endogenous bases. So next is the alkylating agents. One group is called the nitrogen mustards. Just for some interesting history, I have up here that the nitrogen mustards were used in World War I by the German soldiers in the form of a gas, and this gas was actually used to burn the skin and the lungs of all the enemy troops. Okay, so this is interesting that basically might consider the Bible of pharmacology that's edited by Drs. Goodman and Gilman, the one we use here at Wilkes University. Well, these two guys, when they were at Yale, they confirmed that nitrogen mustards had anti-cancer activity. So we actually took something, or they took something that was very toxic, and they determined that if given at the right dosage, it doesn't have to be a fatality, you could actually reduce the cancer burden. Of course, these drugs are extremely toxic, so they're still going to be used, as sparingly as possible. Okay, we'd rather use more specific agents. Okay, the reason why they called it mustard gas, they claim, if you read about it, different sources claim it has an odor like mustard, some of the impurities in the gas, and also causes mustard-colored blisters. Okay. So if you look here on the right, I'm showing you the mechanism by which a nitrogen mustard gets activated before it binds to DNA, and that's exactly what it does. It'll bind to DNA once it reaches its final point. So all nitrogen mustards have some kind of R group, an alkyl group here. Several different types of groups can be there, an amine, and then a chloroethyl. So 2 chloroethyl groups. And the importance of this is that the chlorides are, just like all halogens, are pretty good leaving groups. So if you remember from organic chemistry, a leaving group is something that'll very easily come off, and it makes, kind of gives this molecule incentive to undergo an intramolecular reaction. So the amine literally can attack this carbon that's bound to the chloride group. Okay, chloride comes off. We have the cyclization here in the ammonium ion. That very rapidly gets converted into a carbocation or carbonium ion. That carbonium ion could bind to guanine. This is guanine that's already in the DNA. This carbonium ion could bind to that amine there, and that's a covalent bond. So what will happen is you're permanently modifying the DNA structure. Okay, and that hopefully will induce apoptosis in cancer cells. Sometimes you actually have guanine from, guanine in this bond here that could actually get cross-bridged. So you could have this carbonium ion forming here, and the carbonium ion forming, then you could actually have it on the other side as well. So you have a double one, and you could form a cross-bridge between this amine and this amine. Okay, and you could form, on one strand, you could form an adduct between 2 guanines. And essentially what happens is we get the guanine that's modified. When the DNA tries to undergo replication, that guanine then mispairs with thymine. Okay, so that's not normal, right? It should be ATGC. Guanine mispairs with thymine. Okay, and then the problem is every single strand after that was made with this thymine-containing strand then will contain the wrong nucleotide, and that will be adenine. Because you have adenine and thymine in a place that it should not be. Of course, and that leads to, that hopefully will lead to our DNA checkpoint proteins recognizing an abnormal gene and maybe destroying that, okay, or undergoing apoptosis with the cell, okay. And it's actually really important to understand that in order for apoptosis to occur efficiently, we need a functioning p53 protein. So if a patient has a mutation in p53 rendering it dysfunctional and non-functional, and then we try to use an alkylating agent, we may get lower efficacy than we would if they did not have a p53 mutation. Okay, there's a lot of alkylating agents, a lot of nitrogen mustards. No reason to get into all of them in this course. Most common one is cyclophosphamide. This is probably a pretty old image, but that's what it looked like in the vial there. Cytoxan, brand name. Okay, cyclophosphamide is the most common nitrogen mustard. It is a prodrug, and this is one of the most important things for you guys to remember when it comes to the exam and when it comes to if you do any oncology rotations or for your career. Cyclophosphamide has two major pathways. It can go to the active phosphoramide mustard all the way on the right here, or it can go down here. Now these are a lot of inactive metabolites, but the chloroacetaldehyde in the very bottom left is highly nephrotoxic and neurotoxic. That is not considered therapeutic. At least the evidence indicates that it doesn't really help with killing cancer cells, but it seems to be very neurotoxic. Okay, so ideally we would only go to the right and avoid this toxic metabolite. Okay, but that doesn't happen. We get a combination of those two things. Now we have a lot of enzymes working up here on the top. I really want you to be primarily familiar with CYP2B6, the fact that that's polymorphic, or it could be, you could see drug interactions on there. And now it's just about logic, realizing that if we inhibit this, or if a patient has a non-functioning CYP2B6, realize we have less active metabolite, and then we'll go down and we'll actually get more neurotoxic metabolite, this chloroacetaldehyde metabolite. Unfortunately, though, when we do produce the active metabolite, it's a 1-in-1 or 1-to-1 ratio between this phosphoramide mustard, which is therapeutic, and the acrolein. Acrolein is not considered therapeutic metabolite. This is actually quite toxic, and because of its chemistry, it targets thiol groups very strongly. So any cysteine in your bladder or your renal or kidney proteins will be targeted. And we're talking, any cysteine, so it could be in a drug transporter, it could be in a channel, it could be in a receptor. Okay, anything on the membrane of these cells in the kidney or the bladder that has cysteine will potentially conjugate with acrolein, and that will actually render the protein dysfunctional. So if we have all these channels and transporters on the membrane dysfunctional, eventually the cells will die. So we do get bladder damage. Hemorrhagic cystitis or bleeding bladder can happen. Okay, Mesna is our antidote here. This will save us. Okay, so Mesna is, as far as I know, it's given every time you're going to take cyclophosphamide, because Mesna does have a thiol group, SH, and that serves as a suicide binder. So rather than binding to our endogenous proteins, we'll bind acrolein with Mesna, and then hopefully get eliminated. Okay. So these are the polymorphisms. Again, CYP2B6. Okay, and there was one study that showed that only 56% of patients had the common genotype, while other patients either had one polymorphic or two polymorphic alleles. So it's extremely polymorphic. That means a lot of patients respond differently to cyclophosphamide. 3A4 is polymorphic, but probably more importantly, we have just a seemingly endless list of drug interactions on 3A4. Okay. So we do have genotyping or metabolic activity tests for these enzymes as well. Okay, and I have a question here that we answered in class. Okay, and hopefully you guys can get this. If a patient is on several drugs that inhibit CYP3A4, that same patient is homozygous for a highly active polymorph of CYP2B6, what are the expected consequences? Okay, well that's actually quite easy. If you know this pathway, CYP3A4, of course it's up here, but down here it's really the only enzyme with A5. These are the only enzymes that can produce the the toxic metabolite. So if you were inhibiting this and you upregulated CYP2B6, most likely you'd get more of the phosphoramide mustard, less of the chloroacetaldehyde, but also you get more acrolein, and that's the key point here. Now this is more of a kind of an academic or theoretical situation. We're going to have to consult Dr. Manning and maybe some references to make sure. Essentially, what I'm saying is administering more Mesna might be the best option if you know you're going to be producing more acrolein, or giving a lower dose was suggested in class, and that's probably the most common sense is giving a lower dose, probably the best option. Okay. So I said, what would you do to prevent or to protect the patient? That would be Mesna. Okay. And this is just a little more detail on Mesna. Okay, there's not a lot I want you to understand here. It's probably really good for you to understand that it has a thiol group on it, and that is substituting instead of our cysteines binding to acrolein, the thiol will substitute, and then we'll have acrolein binding to that, and then getting eliminated. Okay, it's very important to understand, though, like I have right here, that Mesna basically concentrates in the bladder, so we have a lot of bladder protection, but the acrolein that gets in the kidney is not really going to be neutralized, so we will still get kidney damage when we use Mesna, because the acrolein is still going to be there by itself, free to bind to cysteines. So the bladder is protected. We should not get as much hemorrhagic cystitis, or we should avoid it completely. But kidney damage is certainly a possibility with cyclophosphamide, regardless of Mesna administration. Ifosfamide is just another example. The only thing I really said in class was that it's going to work the same way, except the metabolism is slightly different. We have CYP3A4 playing the predominant role up top, and 3A4 and 2B6 are both kind of important on the bottom here. But the main take-home point with ifosfamide is that we definitely get more chloroacetaldehyde per milligram we're giving of dose. That means per milligram we're going to get more nephro and more neurotoxicity for ifosfamide compared to cyclophosphamide, and that's probably why cyclophosphamide is the most common drug used in this class. Okay, so it's in my opinion, it's very important for you to understand how the metabolic pathway is different between cyclophosphamide and ifosfamide. Primarily the enzymes that I have emphasized here: 3A4 top and 3A4 2B6 on the bottom. Okay, but acrolein is also formed from ifosfamide. We could also use Mesna to block or neutralize that. Okay, so we're going to continue with, we'll say, quote-unquote alkylating agents, because these platinum-based drugs are technically not alkylating because they don't contain carbons. They're not alkyl groups, but we can call them that. They're generally placed in that category. It's the same story over and over again. The primary function of the platinum drugs is to bind to DNA, cause apoptosis of cancer cells. Okay, we have the same kind of idea here. We get DNA lesions, and they're actually similar for all the platinum agents. So there's cisplatin, carboplatin, oxaliplatin, it's escaping me the other ones, but we have several platinum drugs. Cisplatin is the prototype. That actually has more nephrotoxicity than the other ones. I didn't mention that in class, so hopefully people will be listening to that as well. I probably won't test on that, but it is probably pretty useful to know that cisplatin is the earliest one and is the most nephrotoxic, and there's basically better options these days. So what we see again is guanine intrastrand adducts, adenine guanine intrastrand adducts. So those are the highest percentage, and that means on the same strand we have adjacent nucleotides getting stuck to each other covalently, and that modification in the DNA should cause apoptosis. Something I didn't mention in class. I don't want you to memorize it, but I would like to explain this to make it clear that this large blue protein here is the theory, and that what happens theoretically is that, and there's some evidence, good amount of evidence for this, but the platinum drug, cisplatin, will bind to the DNA, and that'll actually all of a sudden create affinity for a protein that's not normally there. Okay, and this is called the high mobility group protein. You don't need to know that. But once this protein binds, the cell appears to recognize that whole kind of conglomerate, protein, DNA, and drug, as abnormal, and it should commit suicide or undergo apoptosis. Platinum drugs have these leaving groups again, just like the alkylating agents. The theory here is that the their cisplatin is activated by water. There is a theory that says basically that cisplatin is activated to a higher extent intracellularly because they're lacking chlorides intracellularly. So in order for this chloride to be a better leaving group, the theory is that we want to have a lower concentration surrounding this platinum drug. So the theory is that, like in the plasma or extracellular space, there's a lot of chloride, so platinum is less likely to lose its chlorides. Inside the cell, it's theoretically more likely to lose its chlorides, so it appears to have more activity once it gets inside a cell. Because it is possible it would bind to things other than DNA, like a lot of these chemicals can do. But what will happen is water would knock off the chloride, okay, and this will eventually form a positive charge on the platinum. That positive charge gets attacked by the amines in the DNA, and that forms a covalent bond. Okay, switching gears. Now we're still, for this entire lecture, we're going to be on DNA modifying agents, but we're switching to a somewhat different kind, and that would be the topoisomerase inhibitors. I was quizzing the students in class to see if they remembered what DNA gyrase is, because to me it's quite interesting, because DNA gyrase is basically the analogous enzyme in bacteria to topoisomerases in humans, mammals. So DNA gyrase is important for modifying the DNA structure or topology. And believe it or not, the fluoroquinolone antibiotics inhibit this. So fluoroquinolones are kind of like topoisomerase inhibitors, but that's for bacteria, right? They don't really do that to our cells. But our topoisomerase inhibitor drugs, they do inhibit the topoisomerases in our cells, okay, and they're going to interfere with DNA. Well, I'll talk about it in the next slide, but the DNA modifications are going to be basically interfered with in the middle of the procedure. So I'll talk about that in a little bit. So Topo2, it might help you to remember that Topo2 causes double-strand DNA breaks. Topo1 causes single-strand DNA breaks, okay, which is right here. The difference is that Topo2, this is physiological, this is normal. Topo2 causes double-strand break, and the purpose of that is that so we could condense our DNA into chromosomes before cell division. So basically, this is an important enzyme to condense that chromosome. Topo1 is the exact opposite. It creates a single strand break in the DNA, and it actually relaxes the DNA rather than condensing it. It relaxes the DNA in order to undergo DNA replication. Okay, so it's the opposite, but it's a pretty simple concept. Here's Topo1 with a single strand break, or NIC they call it. Okay, now imagine as soon as it creates this break, our Topo1 inhibitor would come right down and bind to it and block it. So it's going to prevent Topo1 from resealing the DNA or religating the DNA. So we get stuck with broken DNA. The same thing happens over here, except it's double-strand breaks. So the DNA never gets repaired, and eventually you'll just have damaged DNA and poor condensation, poor DNA replication, and hopefully cell death for the cancer cells. Okay, first I'm talking about the topoisomerase II inhibitors. Doxorubicin is one of the most common ones. Etoposide may be used less commonly, but it's also considered a Topo2 inhibitor. I always like this word. This is epipodophylotoxin, and so it's in that class. Etoposide is actually taken from the mandrake plant. This is obviously a cartoon image from the Harry Potter series. Okay, that was one of my favorite scenes in Harry Potter. Okay, and then we have doxorubicin, which is considered an anthracycline drug. So there's doxorubicin and donorubicin as anthracycline drugs. Some people call it the Red Devil or the Red Death. And if you know anything about kind of nomenclature in other languages, rubicin is going to be standing for this red color. Okay, so it is literally red in the bottle as it's being administered to a patient. And I think people laughed at me in class because I said that when I look at this red color, it helps me remember the most common toxicity of doxorubicin, and that is cardiotoxicity, because you think of the heart pumping blood, at least I do think of that and make that connection. So maybe that'll help you guys. Hopefully that'll actually be of use to think of it that way. Okay, so the reason why I have this underlined about toxicity is because that's one of the most well-known toxicities of any drug, really, especially anti-cancer drugs. You don't see a lot of drugs causing cardiotoxicity, but doxorubicin is very well known for that. It's actually a little complex because there's an acute toxicity that appears to occur by a different mechanism than the chronic toxicity. I'll talk about that in a minute. Okay, this is actually the acute toxicity right here. We're talking, maybe days to weeks. So acute toxicity is quite a short period of time. The chronic toxicity could be literally years, and I'll get into that in a minute. Okay, but the acute toxicity is believed to occur by free radical formation. So any chemical that has this quinone group with the two oxygens, such as anthracyclines like doxorubicin, any chemical that has that is very likely to undergo this reaction where it's converted to a hydroxyl group, as you can see on the right here, this hydroquinone, and it does that by NADPH CYP450 reductase. That generates as a byproduct the superoxide radical anion, and our body has superoxide dismutase. It's an important protective enzyme that at least detoxifies this radical, produces hydrogen peroxide. Now the problem is hydrogen peroxide is toxic also, but most of the cells in our body have catalase, which will convert that to water and oxygen, and that's healthy, or that's safe. However, the heart has a very low level of catalase, either low or basically none. And what will happen is if we have this accumulation of hydrogen peroxide in the heart, in the presence of iron, which is of course in our blood, we have this thing called the Fenton reaction. The Fenton reaction, you could remember that easily because it's iron with Fe. So Fenton reaction is iron reacting with hydrogen peroxide produce this hydroxyl radical. So this hydroxyl radical here on the left of these three is considered one of the most reactive free radicals you can have in your body, and is very cardiotoxic. We have a drug called Dexrazoxane or Zinacard. Okay, try to pronounce that one 3 times fast. This drug, it does chelate iron, and I mentioned in class that when Dr. Lee, who actually was, for a while, he was with the NCCN helping produce guidelines for cancer treatment. So he's pretty, well established in this field. He said in class a couple of years ago that Dexrazoxane really doesn't have much evidence that it's effective for reducing the cardiotoxicity of doxorubicin. And I'm guessing that's because this is only the acute toxicity. The chronic toxicity occurs by a very different mechanism. Okay, I'm going to skip this because I already mentioned the acute toxicity. Chronic anthracycline toxicity is totally different. The theory, and there's a good amount of evidence to support this, is that calcium accumulates in the sarcoplasmic reticulum. So somehow, if you have doxorubicin, we're talking months to years. If you have enough of doxorubicin, there's evidence that it can permanently increase the loading of calcium into the sarcoplasmic reticulum, the cardiomyocytes. And what will happen then is that calcium gets released through an action potential. You have a very strong contraction, and you could ultimately get hypertrophy of the cardiac muscle. The hypertrophy is very evident in animal models, and we know in humans we get a reduced ejection fraction and heart failure. Okay, what I have underlined here is absolutely critical to understand that doxorubicin chronic cardiotoxicity is going to be completely dependent on the cumulative dose. Okay, so that means not not how much, what concentration you have in your bloodstream at any given time. It's it's not something that reverses. So what that means is that if I took so much doxorubicin on this day, even if I let it leave the body and then I take more a week later, that toxicity is adding up, right? It's cumulative. So over your entire duration of treatment, you should not receive more than 550 milligrams per meter squared of doxorubicin. So yes, it's dosed based on body surface area, not on body weight. And I mentioned in class that's probably related to it not actually distributing preferentially into different compartments. It appears to be independent, right? Independent of fat and muscle. It seems to be more important your actual body surface area. Okay, so there's evidence that there's less than a 1% chance of cardiac toxicity if your cumulative dose is less than 550 milligrams per meter squared. That means from day 1 to the final day of treatment. Okay, if your cumulative dose goes up above 560 to 1155, you can get a 30% chance of cardiotoxicity. Of course, the 30% is probably going to fall in the middle of this range at some point. Okay. Okay, so I have a quick question that we asked in class here. This is something that in the past I think the students were a little bit more prepared for because I talked about so many resistance mechanisms early in this course. But this time you're going to have to rely on your knowledge probably more from other classes that I taught and maybe some other faculty. So the question is, if some investigators have suggested inhibiting MDR1 or P-glycoprotein in order to increase exposure of cancer cells to anthracyclines. My question is, how do you feel about this strategy? So what I'm saying is, we know that P-glycoprotein or MDR1 is in cancer cells. It's a transporter on the membrane. And that's where it was first identified, in cancer cells. That's why it's called multi-drug resistance protein. Now, if we block that, any drug that is a substrate, because this protein pumps drugs out or effluxes them, any drug that happens to be a substrate will then get stuck in the cancer cell. So that might be a good thing, right? It might actually kill cancer cells, and that's very logical. But the problem that I brought up is that, and this is well known, that P-glycoprotein is expressed in the blood-brain barrier, in the liver, in the intestines, and in the kidney. In every single organ, P-glycoprotein is positioned to remove drugs from the body. So it pumps drugs out of the brain and into the blood, and out of the blood into the urine, out of the blood and into the bile and the liver, and then it actually pumps drugs out of the intestines. So it'll actually reduce bioavailability of an orally administered drug if it's a substrate. So if we block PGP, it's pretty much a guarantee that any substrate is going to reach a substantially higher concentration in the bloodstream. So we'll simultaneously increase the risk of toxicity while increasing potentially the efficacy. And theoretically, if we reduce the dose and did this, and then gave an inhibitor of MDR1, we might be able to actually cause more, higher cancer cell or anti-neoplastic activity and less toxicity theoretically. Okay. Topoisomerase 1 inhibitors. We have camptothecin, irinotecan, and topotecan. Camptothecin was originally isolated from the Chinese tree Camptotheca or Camptotheca acuminata, 1966. This was poorly water soluble and severely toxic, so they developed newer compounds based on this. Irinotecan is one of the most commonly used ones. Okay, again, we're talking topo 1 inhibitors, so they're going to block the religation of single-stranded DNA breaks that are caused by topo 1. Okay, irinotecan is a prodrug. It's critical to know that. Okay, and it's also, well, that's important, but I should say that it's actually more important to understand what happens with the metabolite. So SN-38, and I don't remember why it's actually called SN-38. There's probably a good reason in the literature. But SN-38 is the active metabolite of irinotecan. If you bear with me, I'm going to go to the next slide. Now, this is irinotecan's metabolism. Irinotecan is given IV, and it's inactive, but it very rapidly becomes metabolized. It gets metabolized to SN-38 by a serum carboxylesterase. So you do have metabolic activity in your blood, believe it or not. Okay, so it's not just liver, kidney, and other organs, but the bloodstream has metabolic activity, and esterases are very common in the blood. So irinotecan gets broken down into the SN-38 metabolite, which is very active. There's something called UGT1A1, a uridine glucuronosyltransferase 1A1. This enzyme is responsible for converting SN-38 into an inactive metabolite, and then actually the glucuronide that it forms is eliminated much more rapidly. So that means, if you have low UGT1A1 activity, you will accumulate the active metabolite, and you will actually get more toxicity. Okay, maybe more efficacy, but absolutely more toxicity, which is quite worrisome. Okay, switching to the next slide. Something that you need to focus on very carefully here. I'm going to talk more about pharmacogenomics, this time with UGT1A1. What I want to highlight here is most of the pharmacogenomics we talk about would be in the coding region of the DNA, which I have in blue down here as an example. The coding region is the only part of the DNA that makes the RNA, okay, and that RNA is used to make the protein. So the only way you get a mutation in a protein is if you have a mutation in the coding region, okay? But UGT1A1 is different because there are no mutations or no common mutations in humans in the coding region altering its function. Okay, so people that have a mutation, they have the same exact amino acid sequence as someone who doesn't have the mutation. That's because the mutation is in the promoter region. Okay, the promoter region, I hope you had this in Bio 121, and I hope you remember it. But the promoter region in DNA that's the piece of DNA that's not transcribed into RNA. The sole purpose of the promoter is to determine the rate at which the coding region is transcribed. So it's like the gas pedal, right? So if we have a mutated promoter, right? If the UGT1A1 promoter is mutated in this DNA, that is kind of like putting a block of something under your accelerator. You could not accelerate to the maximum potential. So that means we're going to be driving less transcription. We'll have a lower transcription rate of UGT1A1. So patients with this common mutation do not have altered UGT1A1. They don't have an altered protein. They just have a lower concentration of the enzyme. So lower concentration ultimately means lower function, right, overall. So it's the same consequence, but it's a different way of getting there. Okay. And there's a little bit more detail on there if you'd like to read that. I mentioned in class you don't have to memorize the specific number for this mutation, but just realize the patients at greatest risk have a mutation in both alleles. So both promoters in the chromosome, both promoters of UGT1A1 can be mutated to this asterisk 28 form. And these patients have weak promoters in both copies. Patients with this dual mutation, believe it or not, even though it's a dual mutation and they're producing less UGT1A1, we don't usually see toxicity or extra toxicity unless they're in the highest or in the higher side of the dosing range for irinotecan, right? So we need to actually be a little bit higher in the dosing range. So you don't always see toxicity if you have this mutation. It's not like TPMT, where it's pretty much guaranteed. UGT1A1, at a lower or medium dose, you may not see toxicity even if you have that mutation. Okay, finally, we're in the home stretch here. I want to talk about something totally different. We're still on DNA modifications, but this is called epigenetic control of gene expression. I believe this concept is foreign to just about every one of you, and believe it or not, it's foreign to most pharmacists probably, and a lot of scientists haven't heard this term, but it's actually quite important. Epigenetics, what that means is that's kind of outside of the gene or outside of genetics. It's not exactly, it's not a perfect translation of that. But what this refers to is that, we know we can control the DNA or the protein sequence if we modify the DNA sequence. We know that mutating the DNA can cause changes in function, but there's a lot of evidence now that after the DNA gets replicated, it could be modified. So we're saying even if the sequence doesn't change, we could covalently modify the DNA after it's produced to then modify its function. This is called epigenetics. The two most common modifications are methylation and acetylation. Okay. Okay. So methylation occurs directly on the DNA. Okay, and that always results in reduced gene expression. So if a specific gene is methylated, literally methyl groups binding to the DNA through an enzymatic reaction, that results in reduced gene expression, reduced transcription. Acetylation is a little different because we don't acetylate the DNA directly. What happens, if you remember what histones are, okay, that's the protein around which the DNA is wound. Okay, so basically, that's the chromatin. So you guys probably, I hope you remember what chromatin is, but what that is it's a long-stranded DNA that's wound around histones. And all histones are kind of like a ball of protein surrounded by DNA. And what happens, it turns out that a lot of that is for regulation of the DNA transcription. So we could acetylate a histone, or we could hypoacetylate or hyperacetylate a histone, and that'll affect gene expression. And I'll talk about how that happens in a little bit. Okay, some of this down here is just kind of trivia, but I think it's quite interesting. A lot of people consider epigenetics one of the most important reasons for why we have different organ development, because of course the DNA is the same in all the cells, right? So what actually makes everything look different in the body? What changes the function of all these organs? And that's basically epigenetics. So if there's the same genes in every single cell, why do the cells look different? And one reason is what we could actually shut genes off, turn them off if we don't want them to be activated. Through methylation, we could shut them off. We could turn them on through acetylation of a histone. I'll talk about how that happens in a little bit. Okay, we have drugs then that can modify these processes, hopefully to upregulate tumor suppressor proteins. Okay, so this is the example with acetylation. Okay, we're kind of getting from microscopic to more macroscopic. We have DNA, then we have our histone, which is represented as this blue sphere here, and then we have the DNA wrapped around the histone. Okay, if we zoom in on the histone, we might see some lysines. We'll definitely see lysines, and that's the example down on the bottom here. Okay, so some lysines will have an amine. They all have an amine, but some lysines will be charged with a positive charge. Some of them will not be because they'll have the acetyl group, right? Acetyl group bound to the amine, and this is a critical step here. What happens basically is that when we have a free amine with a charge on it, positive charge, that very strongly binds to or pulls in the negatively charged DNA. Because of the phosphate group on the DNA, it's negatively charged, pulls in the negatively charged DNA, and it locks it very closely to the histone. This way, when the DNA is so tightly wound around it, we cannot get RNA polymerase to function. Okay, so RNA polymerase is normally going to bind to the DNA and then transcribe the RNA, but the RNA polymerase won't function. Okay, if the DNA is too tightly bound to the histone. Okay, so that means hypoacetylation or low acetylation means less gene transcription. However, if we acetylate this amine on lysine, we'll remove the ionic bond because there's no more charge here. So the DNA, which is, and just to clarify, the DNA is not shown in this image right here on the bottom, but what will happen is the DNA will no longer have affinity for this amine because there's no more positive charge. The DNA will relax. It'll give space, just steric space, for the RNA polymerase to come in and synthesize RNA. Okay. We have drugs that can influence HDAC, and I'll talk about that in a few seconds. Okay. First, this is just an example of methylation. The one thing I probably didn't mention is that if DNA is methylated, it occurs through an enzyme called DNA methyltransferase. Okay. Up here in this box is one example of what can happen. In some cancers, we have methylation causing reduced expression of a tumor suppressor gene like P16. That could cause cancer because we're reducing the expression of an anti-cancer gene or tumor suppressor, so that causes cancer. Okay. One drug we have as an example is Vorinostat. That's relatively new. Vorinostat is an HDAC inhibitor or histone deacetylase inhibitor. If we immediately focus in down here, if we block HDAC, look at this arrow right there on the bottom. If we block that function, we know we're going to have more acetylation, right? Hyperacetylation of the histone, right? And when that happens, the DNA relaxes, and then we're able to get more DNA transcription into RNA. Okay, and to tell you the truth, there's no proof that Vorinostat specifically upregulates any particular gene. The hope is that the tumor suppressor genes are affected or upregulated more than the other genes. Okay, and that might be the case because a lot of the tumor suppressor genes are preferentially inhibited in cancer. So hopefully we could upregulate that to a higher extent than other genes. Okay, and one example would be p21. Okay, upregulating that and hopefully causing retaining or getting back our tumor suppression ability. Okay. Now we have the other guy, that's Azacitidine. That's different because that's going to be a DNA hypomethylator. So we have the DNA hypomethylator here, and we just talked about a histone deacetylase inhibitor. So one easy way to think about this is both of these drugs upregulate transcription. They just do it by different mechanisms. Azacitidine and Vorinostat both upregulate transcription, and the goal is to upregulate transcription of the tumor suppressor gene. Okay. Now, I'm going to skip this quickly because it's easier to describe on the next slide. Oops, right here. The theory by which azacitidine works is that, let me start at the top. And show you how methylation occurs without the presence of a drug. Okay, this is methylated DNA. It may start off methylated from a previous methylation reaction. The DNA gets replicated. It doesn't immediately have a methyl group on it. It has to be methylated again by DNA methyltransferase. So that's normal. So we have the methylation, replication, and the methylation again to make any daughter cells or progeny to have the same methylation status as the original cell. If we have azacitidine down here, what will happen is we'll get the methylated DNA, okay, undergoing replication, and when it replicates, we might have incorporation of azacitidine, so we have an artificial chemical being incorporated into the DNA, and DNA methyltransferase will still try to come down and methylate it, but there's strong evidence indicating that this DNMT, or methyltransferase, gets permanently locked onto the DNA because of its interaction with azacitidine. So if we do this enough, we will sequester or bind all the DNMT, and then subsequent DNA replications will actually have no methyl groups because this enzyme is rendered non-functional. So then we're actually we're hypomethylating in that case. Okay, this is a continuation. So 1, 2, 3, 4, 5, 6. So we ultimately continue this process and get unmethylated DNA. Hopefully that upregulates the tumor suppressor gene. Okay. Again, now the three possibilities are sequestering a DNMT. Okay, and when that happens, there is evidence that this structure can cause apoptosis. There's evidence that azacitidine in the DNA by itself can cause apoptosis, and this is one of the more interesting points, that using azacitidine can, in some cases, it's been shown that it can take cancer cells and cause them to revert back to a normal cell. So this is a rare thing. We usually don't see this with drugs, that you might be able to take a cancer cell and make it turn back into a normal cell. Okay, and no one's really said how likely this is, how common it is, but there is evidence that this has happened with this drug. Okay, so we want to hopefully upregulate tumor suppressors, suppress cell division, potentially transfer the cell or transform it back to its differentiated state instead of the de-differentiated state. Okay, for this critical thinking question, we went through this, I think, very quickly in class. Question is, what is a potentially major detrimental consequence of non-targeted DNA hypomethylation? Two of these are pretty simple. 1 and 3, you should not pick those because hypomethylation causes overexpression. So we can't have reduced expression, so we should have increased expression. That's the first point. But now let's look into this further. So increasing expression of growth-promoting genes, can that happen with DNA hypomethylation, and is that detrimental? Yes, it can happen because these are not targeted agents. Azacitidine is not targeted. And of course, we don't want growth-promoting genes upregulated or their transcription upregulated in a cancer patient, right, or tumor. The other side, though, is can we get increased expression of a pro-apoptotic gene? So that's the opposite. So more apoptosis. The truth is, yeah, we want apoptosis of the cancer, so that's a good thing. Of course, the problem is that azacitidine doesn't only bind in cancer cells, it binds to DNA in healthy cells as well. So, this is a little tricky because it is therapeutic, but it's also detrimental to have apoptosis in, non-specifically in all cells. So that's basically the answer. The answer to that is B, 2 and 4. Okay, and actually I keep forgetting that that's already in your handout, but it's animated right here. Okay, and it looks like that wraps up the presentation. It's in about an hour and 2 minutes, probably slightly quicker than, actually a lot quicker than it was in class. This is my first time ever doing a presentation like this, podcast-like presentation. If you have any comments or questions, please let me know. If you think it was good quality, bad quality, if you think it was beneficial, any frank feedback would be greatly appreciated by me. So hope you guys get time to look at this soon, and but depending on when you hear this, have a good day or have a good evening. Okay.